Okay, thank you very much for giving me the opportunity to give the talk today. So I'm coming from the other side. So we develop the test systems and deliver the data before the bioinformaticians start to calculate. And about one of our currently developed test systems, I want to show you the results, or better, I have to say, the problems that we faced. So I guess we all agree that we have current large issues in toxicology. Okay, we have a huge backlog in testing. There's a lot of chemicals that are not tested. Therefore, we need faster test methods. And further, there's in some areas there's a ban of animal experimentations, for example, cosmetics. So anyway, there we need new technologies. We all know that there's a low predictivity of animal experiments, especially in the developmental toxicology field. So there's a better human predictivity needed. So in 2008 there was this Tox21 agreement to have a paradigm shift from in vivo animal to more in vitro assays. So what our lab is dealing is with developmental neurotoxicology, and basically we had three challenges. So it's the first one is the availability of proper test methods, and in developmental toxicology you have a changing baseline, which has an influence on timing of treatment. Further, the in vitro, this is a problem or challenge of in vitro test systems. What is the functional consequence of all the data that you generate? So especially during early development or early neural development, the closure of the neural tube is a very sensitive time window. So there's one famous compound that is valproic acid, and this is only teratogenic during this specific time window of embryonic development, and it leads or increases the risk of neural tube defects such as spina bifida or anencephaly. So therefore VPA is our favorite tool compound to test our systems. So in more details, there are many processes that are involved in neurodevelopment. So our first step is lineage specification towards ectoderm and neuroectoderm. The neuroectoderm then builds the groove, the neural groove. This groove closes to the neural tube, and the cells that build up the neural tube, they are called neural epithelial progenitors. These progenitor cells undergo further lineage specification, either to clear progenitors that develop into mature astrocytes, we are not interested in that, or neuronal progenitors that mature into the different types of mature neurons. So these later processes you can monitor by neurite outgrowth assays, synaptogenesis, measuring the cell death, transmitter turnover. We have some test systems developed in the lab that models exactly, so neurite outgrowth, for example, or cell migration. But today I want to show you a test method that we have developed and is called UKN1, and it's based on gene expression analysis, the change of gene expression patterns. And UKN1, that is because it's our first test system that we developed, and its UKN is University of Constance. So we did not have a lot of fantasy on the name. So the biological system is based on human embryonic stem cells, and in the meanwhile we can also do it with induced pluripotent stem cells. It's based on a dual SMAD inhibition by noggin and a speed drug that prevents the differentiation to the other germ layers. It pushes the cells into the ecto-neuroectoderm lineage, and at the end, after 14 days, we end up with a quite homologous population of neuroectodermal progenitors. So that over 90% are PAX6 positive and nestin positive. So we have further characterization, but this is due to time limitations. I did not show all the stainings and quantifications. There is a little characterization by gene expression analysis. So as expected, the pluripotency markers as NANOC and OCT4 are strongly downregulated, whereas neuronal markers are upregulated. So together with a whole transcriptome analysis of the Affymetrix platform, we can say that the cells correspond to a rostral localization in the neural tube around neural tube closure. In 2012 we published our first study on this test system, and of course we used VPA and treated our test system, and what we found that if we treat it for 6 days with VPA, we end up in different or in wrong neuronal precursor cells. This was identified by transcriptome and epigenetic markers, and we did in this paper a very extensive qualitative characterization of gene expression changes. But in a toxicological point of view, it does not help us very much. So genes are changing, and yeah, so what? What we need is quantification, and the big question after 2012 arose, how can we quantify gene expression changes based on transcriptome changes? So therefore we started a concentration response study, again using increasing concentrations of VPA, and what you see here is a normal viability concentration response curve based on the Rosette. And you see in green we have non-cytotoxic concentrations, and starting after around 550 micromolar the cells start to die. So for each of these concentrations we did an Affymetrix whole transcriptome analysis, and in the first step very simply we counted the significantly differentially expressed probe sets, and you can see here nicely that starting with 350 micromolar VPA the genes start to change. In red you have the cytotoxic concentrations, but already at these concentrations with 350 micromolar we start to have an effect on gene expression level, which we did not have on viability. So therefore we can define, or we assumed a third concentration range, namely maybe this is the one that we are interested in developmental toxicology. And this is also exactly the range that you find in plasma, in human plasma. We also did this, not we, but our colleagues from the bioinformaticians, a principal component analysis, and also on the basis of the whole transcriptome you see a nice concentration response. And the middle concentrations, they cluster together, and the cytotoxic concentrations. So basically here we did a first quantification based on the amount and variance of changed genes. So it gives us a first hint, and also importantly it is the no effect level is different for cytotoxicity and for gene expression. Okay, in the next step, as we are biologists, we tried to put or to bring biological sense into our thousands members gene list. So we did all the databases that were mentioned before, and I just show you the gene ontology database. We put our gene lists into this database and counted again how many overrepresented gene ontologies we get. And this you can see here in the graph. We get starting with 350 micromolar, we get overrepresented gene ontologies, but already 80 here in the first bar you see 80 up to over 100 overrepresented gene ontology. Nice. And we summarize them in, so you find a lot of neuronal development, GOs that deal with neuronal development, of course, GOs that deal with cell cycle are downregulated, but it was still not very usable in a toxicological sense. So also most of the GOs were already overrepresented, and only a few are added with the cytotoxic concentrations. Again, this is more a qualitative description. But then we thought, okay, maybe we can combine that and come up with a quantification. And this is what we call the Teratogenicity index. We calculated a measure for the regulated genes based on the p-value and a measure for the amount of gene ontologies. And you can see here, basically we have the same results, that with the low concentrations that are not cytotoxic, this index is very low, but it jumps up to a basal level with 350 micromolar. So again, what we can say, the more genes are changed, in this case, the more biological processes are affected. Therefore, maybe we can give or make a cutoff, in this case with 350 micromolar, starting to be teratogen. A second approach, or a second idea, is to develop to calculate a developmental potency and a developmental index. So, in the first step, we identified the developmental genes. So the genes that are important in an undisturbed differentiation that are regulated. In the case for UKN1, it was 1,238 genes that seems to be important for development, and we called them D genes. And in a second experiment, we identified the toxicological genes, so the genes that are changed by a compound. We called them T genes. And then we did an overlap analysis, and this is the overlap between D and T. And first we thought, okay, the more developed D genes, developmental genes, are affected by a toxicant, the more development is affected. But this did not give us, so we lost another information, like for the 1000 micromolar, so the overlap increased, but in addition, much more additional genes were added by the cytotoxic concentrations. So we did two measures. So here the overlap is higher, but many additional non-developmental genes are regulated, which is completely logic, because now the cells start to die, they start to induce stress responses and apoptotic genes and so on. But this is maybe exactly that you want to distinguish. Has it an impact on development, or are the cells just dying? So therefore, the developmental potency, that is the overlap in relation to D, and the developmental index is the overlap in relation to T. And this is how it looks like in the mathematical formulas. And in our case, in our concentration response study with VPA, you see here the developmental potency. This is increasing, and it means for 800 and 1000 micromolar that 50% of the genes are in the 50% of the D genes are also changed by treatment. And the developmental index is decreasing. So that means the overlap compared to the tox genes, to the genes that are changed by the compound, is decreasing, because the tox gene only amount is increasing. So this gives us an idea, or a quantification of exactly the effect that I showed you in the slide before. And indeed, if we investigate these tox genes only that are not in the overlap, we find a lot of non-developmental genes, including stress response pathways, for example. So the next issue I wanted to show you is the timing of treatment. We have a special problem in developmental toxicology compared to a stable cell system, that in a stable cell system you have a baseline, and a gene can be upregulated or downregulated or not changing. In a developmental system, you can have a shifted baseline, so that a gene is just not as much upregulated as normal, or not as much downregulated as normal. And you can have have the situation that a gene is only downregulated at the end of differentiation, or in the middle of differentiation, and regenerates and the downregulation is reversible, for example. So therefore it might be important at which time window and for how long a developing system is treated. And this is a simple example here. Here is again a PCA, again the treatment that you have seen before between day 0 and day 6. And if we do the same and treat our cells only the last 2 days, you can see in the PCA, so that the answer, the response is different, so that these are not the same genes, and but genes are changed. And the same, these are also around thousands of genes that are changing, but they are different. And if you do the overlap, they are not the same. Just because we start the treatment 4 days, 4 days? Yeah, 4 days later. So the concentration response is always, and we have some more examples, this is just one example, we did a big time window study, and the concentration response is dependent on the timing. And these are only 3 examples of our big time window concentration study. Here you see in blue so on the x-axis you have the time, and in blue you see the control, the untreated situation, and in yellow, orange, you see the treated situation. So we did it in parallel, treat them different time points for different times, take samples and did whole transcriptome analysis, and these are only 3 example genes that I show you here. So PAC6 is the typical shifted baseline. It is not that much upregulated than it should. OTX2, it's the same, so it normally is regulated much higher. And what is interesting now, and I guess it's also interesting for the modelers, that the genes that peak during differentiation, these peaks are shifting either to the left or to the right. And this is at the moment a project that we do in collaboration with systems biologists in Berlin, and they try to model this effect of these non-monotonic changes. So that means also that each gene has its own baseline, and based on this baseline and on the time point that you treat or analyze, it's better to say analyze, this has a big influence on the outcome. So if you look at the DAZL gene, if you would take, can you see? Yeah, if you would take only this time point here, you would not see a difference. So this is everything is very nice. We thought, okay, yes, genes are changing. We can maybe model it in a collaboration. But still, what does it mean? So, what does it mean when genes are changed at day 6 of differentiation? So we were thinking about a functional analysis. So has it a consequence? Therefore, we tried to do a phenotypic anchoring and further developed our differentiation system. So the normal differentiation system that I showed you until now was always until day 6. Then we further developed the system and differentiated our cells to the next stage of development, the so-called Rosette stage cells. In this stage, the cells migrate to each other and form so-called rosettes, which are considered then to be really the neural tube state, or corresponds to the neural tube state. And the question was, if we treat our cells from day 0 to day 6, do they still differentiate into these neural rosettes? Here you can see a little characterization of these rosettes. They are really beautiful. So we used the ZO1, this is an extracellular matrix protein, and GM130, this is a Golgi apparatus protein, of which we know that it's assembling towards the center of the rosette. So this is a detailed picture, and here you see an overview picture, and each bright dot here represents a rosette. So we did our UKN1 treatment scenario from day 0 to day 6, and analyze the cells then, the rosettes on day 14. And indeed, here you have the control situation. And if we have treated the cells for 6 days, they differentiate into more epithelial-like structured cells. You see here in red the extracellular matrix, so they look more like epithelial cells, and we do not find any rosette in the well. We also developed, or colleagues of us from the informatics department developed an algorithm to quantify the rosettes. And so yeah, you can see the algorithm is working very well. Each dot represents the number of rosettes per well. Okay, so we concluded, at least for VPA, yes, our gene expression changes have really functional consequences in further differentiation. And therefore we tested more compounds, and in the meanwhile it's even more than I show you here. So we used, the first place, more HDAC inhibitors and also unrelated compounds. And here you see the group of HDAC inhibitors. They all impaired the rosette formation, as well as Wnt activators, which is also logic because Wnt is important signaling pathway. Endocrine disruptors like VPA do not impair rosette formation, and mercurials, there are some that do and the others are not. So now next question arose, do this correlate with differentially expressed genes? Really, is that the point? Can we say, okay, if genes are changed, whatsoever genes on day 6, do we then have an impact on rosette formation? And this, we can say no, this is not the case, because if you look here on this methyl mercury, you have differentially expressed genes, but no inhibition of rosette formation. Whereas for retinoic acid, for example, the same amount of genes is changed, but we have a full inhibition of rosette formation. So therefore, what we think and what we are working currently is to ask the question, is there a specific gene expression pattern on day 6 that may predict later rosette formation impairment? And this may also help to further develop the test system and to simplify it. If we end up with a set of genes, let's say 50 or 100 genes, it is much easier to analyze than thousands of genes as an Affymetrix chip. And here I want to come back in the last slide to our timing problem. Remember that we had this treatment from day 4 to day 6. So this is the control situation, this is the control, we have the rosettes, and after 6-day treatment we see the epithelial cells, and we ask whether what happens when we treat from day 4 to day 6. We are also gene expression, we have huge gene expression changes, but still rosettes are formed. So these data sets might help us to identify biomarkers that may predict is exactly if on day 6 if differentiation might be affected. And this brings me already to the summary. So I have shown you that we have developed an early DNT test system called UKM1. I proposed some quantification approaches, quantification ideas. I've showed you that timing of treatment must be considered, and that the formation, the rosette formation as a functional endpoint of the test system. Our open questions at the moment is there this specific gene set that may predict the impairment of rosette formation, and of course is it relevant in vivo? So the in vitro in vivo extrapolation has to be done, or is ongoing, and how can we simplify the test system for broader applicability? Because at the moment you have really to be expert on cell culture to perform these tests. Okay, this brings me to my acknowledgements to our collaborators. So Agapius from the University of Cologne was doing the whole Affymetrix gene shift measurements. Mariana and Jörg, they analyzed it. They are from the University of Dortmund. Johannes and Niels, they are the system biologists that do the modeling at the moment. And of course the team in Constance, Marion and Nadine, they set up the whole system, and Nadine especially developed the Rosette endpoint. And of course, last but not least, our boss, Marcel Leist. And thank you for your attention.